My name is Veronica Chiang, and I will be presenting my research on the analysis of in silico structure-based and ligand-based screening approaches to optimize drug design on the glucocorticoid nuclear hormone receptor. My project was conducted under the supervision of Dr. Shapira in the Department of Pharmacology and Toxicology at the University of Toronto. To provide some background on my protein of interest, the glucocorticoid receptor is a nuclear hormone receptor which predominantly resides in the cell cytosol in a transcriptionally inactive conformation. When activated by ligand binding, the receptor undergoes dimerization and entry into the nucleus, where it binds to glucocorticoid response elements on the DNA and functions as a transcription factor. This receptor is involved in various signaling pathways that are primarily responsible for the regulation of body homeostasis, such as metabolism, inflammation, cell growth, and other cardiovascular and endocrine functions. Structurally, the glucocorticoid receptor consists of three principal domains: the N-terminal transactivation domain, the DNA binding domain, and the C-terminal ligand binding domain pictured here, with a flexible hinge region located between the DNA and the ligand binding domains. Its endogenous ligand is cortisol, which is a steroid-derived glucocorticoid produced from the adrenal glands, and activates the receptor when it binds to the C-terminal ligand binding domain. Due to its prolific physiological profile regarding both the locality and function of the receptor within the body, researching tissue-specific drug compounds that have a high structural affinity and potency to the ligand binding domain, as well as developing non-steroidal agonists and antagonists, is much favored. The objective of this project is to utilize an in silico protein modeling and drug design software to analyze the structural diversity and chemical interactions between the glucocorticoid receptor and compound libraries, as well as between known glucocorticoid receptor ligands and random compound libraries. As a brief overview, I used the Protein Data Bank website to obtain ligand-bound crystal structures that were appropriate for fragment docking. Next, I conducted various computational assessments, ultimately to compare structure-based and ligand-based screening methods, as well as optimize known agonists and antagonists of the glucocorticoid receptor for further drug development. As previously mentioned, the crystal structures and ligands for the glucocorticoid receptor were obtained from the Protein Data Bank website and loaded into ICM Pro by MolSoft. This in silico protein analysis software features many tools used in areas ranging from bioinformatics to computational chemistry and will be outlined in this video. The ICM Pocket Finder feature was used to identify possible cavities for ligand binding on the receptor based on a grid potential map of van der Waals interactions established by the receptor's overall shape. Multiple sequence alignment was conducted to determine structural similarities between the amino acid residue sequences of the glucocorticoid receptor and other adrenal steroid receptors. 3 docking simulations, receptor fragment docking, receptor cross-docking, and a 10,000 fragment library docking, were conducted using a similar approach in ICM Pro. This process involved setting up the receptor and creating receptor maps. Fragment docking of approximately 2,000 lead-like molecules, as well as known ligands, determined interaction hotspots of the glucocorticoid receptor's ligand binding pocket. 4 different crystal structures of the glucocorticoid receptor were obtained from the PDB website and underwent a cross-docking procedure to identify which conformation has the greatest ligand binding capacity. Finally, a receptor-based docking simulation was run using a library of 9,925 compound fragments. A 2D clustering analysis was performed to identify common scaffolds using active glucocorticoid receptor molecules obtained from Kemball's diverse library of 3D compound fragments, including both agonists and antagonists. The structure-activity relationship tool allowed for the comparison of the impact that different R groups had on its scaffold's activity score, as identified from the 2D clustering procedure. The 3D Quantitative Structure-Activity Relationship Tool, or QSAR, was used to build a prediction model using active glucocorticoid receptor molecules from Kemble. This model was then used to predict the activities of another library of molecules. The Atomic Property Field Feature, or APF, analyzed scaffold alignment superpositions with known ligands, using their pharmacophoric profile, which are the steric and electrostatic features of the compound. Based on the compound's atomic field property, the Structure Activity Landscape Index tool, or SALI, quantified activity cliffs between pairs of compounds that were structurally very similar, but had the greatest difference in activity scores, using flexible APF superposition to template. The Rapid Isosteer Discovery Engine Screening Tool, or RIDE, was used to virtually screen large libraries of 1.5 million compounds, using the atomic field properties of known ligands. This tool constitutes the ligand-based screening portion of my project. Lastly, the ligand editor tool was used to optimize known glucocorticoid agonists and antagonists. To compare the data generated from the receptor-based and ligand-based screens, specifically which method had a better chance of success at identifying suitable molecules that could proceed in the drug discovery pipeline, enrichment scores were calculated using this equation obtained from Lee et al. The higher the enrichment score, the better the screening method. A structural diversity analysis of the glucocorticoid receptor was performed using the ICM Pocket Finder tool. This helped to identify any other potential cavities or clefts on the receptor that may be suitable for binding, beyond the typical ligand binding domain. As shown here, the various plausible pockets are depicted in color, with the blue region indicating the original binding pocket of the receptor. 4 additional pockets were identified in total. The data obtained from the multiple sequence alignment showed approximately 14% residue similarity between the glucocorticoid receptor, GCR, and other adrenal steroid hormone receptors, including the mineralocorticoid receptor, MCR, the androgen receptor, ANDR, and the progesterone receptor, PRGR. As expected, the C-terminal ligand binding domain of all 4 receptors contained the most residue similarity, and thus important to keep in mind when designing glucocorticoid-specific drug compounds. The residues that are not highlighted in green are those that differ between the 4 receptors and should be investigated further as potential binding hotspots. This is the alignment of the glucocorticoid receptor proteome and the crystal structure obtained from the PDB website. Since the crystal structure only displays the C-terminal ligand binding domain, no residues appear outside of the red bracket. With a 99% residue similarity match, this confirms the accuracy of the crystal structure selected for further investigation. From a random selection of amino acid mutations in the ligand binding domain, all results obtained ranged from negative 0.11 to 0.17. With a significance threshold of values less than negative 2 or greater than 2, these data indicate that no significant change in binding energy was observed for any of the 6 alterations. Therefore, these residues likely do not play a key role in the ligand-receptor interaction. Instead of performing mutagenesis studies, interaction hotspots within the glucocorticoid receptor ligand binding pocket were confirmed by a docking simulation using a library of over 2,000 lead-like compounds. These hotspots were determined based on the residues that exhibited the highest number of atomic interactions, specifically hydrogen bonds, and are shown here in color. The 4 hotspot residues identified are leucine 563 in blue, glutamine 570 in green, cysteine 736 in red, and asparagine 546 in yellow. Receptor cross-docking was performed using 4 glucocorticoid receptor crystal structures obtained from the PDB website. These were 4P6X, 5UFS, 3CLD, and 1P93. Using a score cutoff of approximately -32, the receptor identified for subsequent receptor docking analysis was 5UFS. Using the 5UFS PDB glucocorticoid receptor crystal structure, a diverse set of approximately 10,000 lead-like screening compounds was obtained from the Life Chemicals website and used for the receptor-based high-throughput screening procedure. This resulted in the generation of almost 3,000 unique binding poses. A closer examination at the top-scoring compounds showed that only 92 of the 29,817 poses had a score of -32 or less. These data will be later used to compare the structure-based screening method with the ligand-based screening method. To begin the ligand-based screening section of my project, a database of active glucocorticoid receptor compounds was obtained from the website Chemble and imported into ICM Pro. Using the clustering tool and with a similarity threshold set to 0.2, 20 scaffolds were identified, each having at least 31 distinct compounds in the scaffold family. The scaffolds with the highest number of compounds, found in Group 3 and Group 14, showed structural similarities with known glucocorticoid receptor ligands, namely the presence of cyclic rings. This outlines the potential importance of ring structures in identifying plausible compounds that would elicit an effect on the glucocorticoid receptor. Based on the scaffolds that had the highest number of distinct compounds, so groups 3 and 14, a structure-activity relationship analysis, or SAR, was conducted, which identified R groups on scaffolds that contributed to the molecule in having the greatest overall activity scores and were most commonly exploited. For Group 3, the R groups that contributed the most were those that contained an ether moiety and an alkene group. For Group 14, these included aromatic nitrogen systems with a carbonyl as well as a trifluoromethyl group attached to a benzene ring. After docking the 20 scaffolds with a crystallized ligand structure obtained from the PDB website, the top scoring or lowest scoring scaffold structures were identified for further atomic property field assessments. These compounds were from scaffolds in groups 10, 11, and 15. Using the same ligand structure as the template, depicted here in purple, the compounds were screened using APF superposition to generate a scaffold molecule binding score graph for trend analysis. Scaffold compounds that consistently have similar and low binding scores would be ideal for further drug development. However, no such trends were seen in any of the 3 scaffold groups, as poor R-squared values were observed in all graphs, 0.03 for group 10, 0.08 for group 11, and 0.01 for group 15. Although the scaffolds themselves scored highly, when various R groups are attached, the binding scores greatly fluctuate. Future analyses that compare the lowest scoring compounds from each scaffold group would be beneficial. Using a random selection of 979 compounds with activity on the glucocorticoid receptor, the atomic property field 3D quantitative structure activity relationship tool was then used to build a ligand model training set for ICM Pro to learn. This then generated a graph containing the activity values, or P-act, and cross-predicted activity values, QSAR, for each compound of the training set. The results generate a small correlation, with an R-squared value of 0.33. This model was subsequently used to predict activity scores for another set of randomly selected compounds with activity on the receptor. Though a small correlation was seen, there was a cluster of data points near the bottom half of the graph, and with an R-squared value of 0.13, this model was not an accurate tool to rank plausible ligands. The Rapid Isosteer Discovery Engine, or RIDE, was used to computationally determine the molecular similarity between known ligands in a library of over 1.5 million compounds in 3D, using pharmacophoric properties determined by APF. 3 known glucocorticoid receptor ligands were used as the template, with dexamethasone and triamcinolone acetonide as agonists, and Ru-486 as an antagonist. Based on the normalized scores obtained from the Chembl database of known compounds with activity, which is the bar in blue, as well as 3 other databases, each with over 1.5 million diverse compounds, the Chembl database achieved a higher normalized score, indicating that a greater percentage of the compounds matched the pharmacophoric properties of the 3 known ligands. This comprises the ligand-based screening portion of the project. The comparison of enrichment between receptor-based and ligand-based virtual screens was conducted using the data obtained from the 10K fragment docking and the Chembl RIDE screen. Based on this calculation, with the equation shown here, the receptor-based screen scored a poor enrichment factor of 0.76, while the ligand-based screen scored an improved enrichment factor of 1.48. This was based on the 10% selection of compounds in the respective screening methods library. Overall, the ligand-based screening method, or RIDE, served as a better and quicker tool to identify suitable compounds that matched current glucocorticoid receptor ligands. With regards to ligand optimization, the residues that line the glucocorticoid ligand binding pocket were first recognized. Using the ICM ligand editor tool, the receptor hotspots identified from the previous structure-based analysis were marked as a starting point for ligand optimization design. The optimization of a glucocorticoid agonist and antagonist, dexamethasone and Ru-486 respectively, was compared. For the antagonist, the region circled in red was explored, as the original ligand normally interferes with helix 11 and 12 in this area of the binding pocket. These data illustrate the original ligands versus the optimized ligand designs and their respective radial and topological convolutional neural net scores, or RTCNN, with lower scores indicative of better predicted receptor interactions. For both the agonist and antagonist ligand, the addition of an iodine atom improved the score. However, a greater decrease in score was observed for the agonist than in the antagonist design. Looking at the binding pocket in coordination with the edited ligands in 3D, with the iodine atom in purple, there is a much favored orientation of the optimized agonist than the optimized antagonist. As the agonist conformation makes a better use of the available space in the pocket. Based on these results, beginning with the ICM pocket finder, there is an indication that the glucocorticoid receptor is a suitable protein for further analysis of protein-ligand or protein-protein interactions. These cavities may be further explored in future drug development studies that involve this receptor. The interaction hotspot analysis by receptor docking confirmed 4 key ligand-binding residues and confirms the significance of glutamine 570 in the binding pocket, as stated in the literature. Most importantly, the comparison between the receptor-based and ligand-based screens indicated that using ligand-based screening methods for drug discovery was a better tool for identifying hits, or suitable compounds that matched known ligands of the glucocorticoid receptor, as a higher enrichment factor was observed. Lastly, using the ligand editor tool, it was found that ligands which were agonists served as better molecules for optimization compared to ligands which were antagonists. Currently, there are more data found in the literature regarding glucocorticoid agonists than antagonists, and thus may explain why agonists are a more favorable area of study for further drug discovery. In addition, researchers face another challenge in the development of antagonistic ligands, since they must consider the helix 12 domain of the glucocorticoid receptor, which must be positioned in such a way to prevent the entire receptor from binding to the DNA as a transcription factor. In summary, ICM Pro by Mulsoft has many beneficial computational tools that can aid in the initial in silico drug discovery process. These tools not only allow researchers to predict molecular interactions and detect key residues in binding pockets, but also help to identify the pharmacophoric profile of known ligands, which ultimately lead to the design of better and more optimized drug compounds. For my analysis, a ligand-based screening method serves a greater advantage over receptor-based screening, and thus should be applied first when starting out with computational studies similar to this one. Further down the glucocorticoid receptor drug discovery pipeline, the development of non-steroidal compounds will be an important avenue of research to explore, especially in the minimization of unwanted adverse effects. Future steps would be to take the compounds identified in the ligand-based screens and run a virtual receptor-based screen to identify those that scored highest. These compounds would then undergo ligand optimization so that better scoring glucocorticoid agonists and antagonists can be developed. I'd like to thank Dr. Shapira as well as the PCL 472 research group for the ongoing support and guidance throughout my project.